Looks like it's 12:40 Eastern. Chris Rogers, you are our session chair for the next session. So I guess
Greg, thank you.
We'll get underway.
Appreciate the opportunity to chair this session. We've got a lot of interesting talks ahead of us. The session's called Advances in Atmospheric Deposition Monitoring. Again, I'm Chris Rogers. I'm with Wood Environment and Infrastructure Solutions. Work on CastNet primarily. All right, so we'll go ahead and get started. Up first is Jonathan Mock with Harvard, and he'll be speaking on the global importance of hydroxymethane sulfonate and ambient particulate matter and the implications for air quality. So Jonathan, you can go ahead and share your screen, and thank you for doing the first presentation of this session.
All right, can everyone see me and hear me? Or see the slides and hear me?
Yes.
Okay. All right. So thanks for having me. As was just mentioned, I'm going to talk about hydroxymethane sulfonate and in ambient particulate matter and implications for air quality. And so including kind of some new modeling work and observations that we have done. So this is kind of the origin of why we got interested in this question. It actually started from a question in China, which was we were trying to understand why was there so much sulfate in these heavy haze events in wintertime? And so basically the traditional understanding of sulfate chemistry is that a lot of it comes from sulfur dioxide being oxidized to sulfate by photochemical oxidants like ozone or OH, and that requires sunlight. On the other hand, in heavy haze conditions, there's a lot of haze that blocks out the sunlight, and so observations of oxidants like ozone and OH are low. And so the question kind of became what was doing the oxidizing of the sulfur dioxide to sulfate. And so this was important because observations from air quality models were showing massive underestimates of PM 2.5 during these heavy haze events. A lot of that was driven by sulfate specifically. So this is an example on the right of figures from the GeoChem chemical transport model, an older version, compared with observations from the US Embassy in Beijing for PM 2.5 in January 2013. And observations reached PM 2.5 of over 500 micrograms per meter cubed, whereas GeoChem got nowhere near that. And a large portion of that underestimate was sulfate. And so the question kind of became, where's all this sulfate coming from? I'm not going to focus too much on China in this talk, but there's an earlier paper that looks more specifically at that. And what we kind of came up with and realized is that there's another sulfur molecule that could have been confused for sulfate in the measurements that also didn't require a lot of photochemical oxidation. And this is hydroxymethane sulfonate. And the chemistry of this is actually very well understood in that it is a pretty simple reaction between sulfite and bisulfite and dissolved formaldehyde. And in general, cold temperatures promote the formation of this because there's more dissolved SO2 and more dissolved formaldehyde. And also important to note is that the formation destruction of hydroxymethane sulfonate, or HMS, as I'll say from here on out, is dependent on pH. So we did some experiments looking at how does this show up in the measurements. And this again was based off of trying to understand initially work in China. And a lot of the work doing measurements of PM 2.5 used ion chromatography with something called an alkanol quaternary ammonium functional group. And it turns out that those type of ion chromatography columns have difficulty or are unable to differentiate different sulfur compounds. And so what I'd like to draw your attention to in the bottom left is this is a experiment using an AS22 type ion chromatography column with, I think it was half, so half of the sample was, exactly half was hydroxymethane sulfonate and half was sulfate. And the peaks kind of got smudged together in such a way that the automatic integration software interpreted the whole thing as sulfate. And so this was what we think was happening in some of the prior measurements of those really high sulfate measurements in China, is that some of it was hydroxymethane sulfonate. importantly, as I mentioned, there's this issue with high pH where HMS can decompose at high pH because of the chemistry, where HMS, if it reacts with OH minus, falls back apart into sulfite and formaldehyde. That means that it can get oxidized in the column. And so for ion chromatography that uses high pH elements such as KOH, that could cause HMS to decompose and oxidize to sulfate in the column. And that can also make it harder to differentiate the two. So this is an example here on the right. This is HMS and sulfate, but at pH 12, and it's even more difficult to separate the two because a lot of the HMS is oxidized and becomes sulfate. Importantly, we found that some functional groups can separate HMS and sulfate. So the example here is from an alkyl quaternary ammonium functional group column. And as you can see here, this is again, half the sample injected on the right was HMS and half was sulfate. And there was a clean separation here, which was nice. So that was good and at a low pH. But there's still this issue of potential HMS decomposition over time. So this is the same kind of half and half sample injected, but at pH 12, and the HMS, sorry, I think I've so this is only HMS, excuse me. And some sulfate starts to appear because there's oxidation of the HMS peak. And so this is just evidence that there are ways to differentiate HMS and sulfate, but there still are some problems. So quickly also with AMS can also have some issues differentiating HMS and sulfate in terms of that HMS itself has no unique identifiable fragments. And so typically what happens with AMS work is scientists, people looking at the spectrum will identify all of the sulfur containing peaks and attribute them all to sulfate. If HMS is there, that can be confusing. And so on the left here, you have a typical sodium sulfate sodium sulfate spectrum. On the right is sodium HMS. And important to note, again, no unique identifiable fragments from the HMS, but basically the HMS can look pretty similar to sulfate and organics. So it's very difficult to tell that it's in there unless you're looking for it. And so again, this just makes it more complicated that you really have to pay attention. There are some methods that are being developed of maybe using the ratio of this SO3 plus peak and trying to use that to determine how much HMS is in there, but there's a lot of uncertainty with that method. So I mentioned before that some traditional HMS methods in China, methods of ion chromatography could not differentiate HMS and sulfate. We have now some recent measurements from a city called Shijiazhuang in China, which is about 165 miles southwest of Beijing. And they using this new ion chromatography system, they were able to differentiate HMS and sulfate and saw a lot of HMS in their samples. And so the black dots here are 12-hour mean observations of HMS from Shijiazhuang. And they saw up to around 7 micrograms per meter cube at peak during a haze event of HMS in the PM 2.5. And using the old system, this would have been identified as just purely sulfate. And so this was a ratio of about HMS being about 15% during the peak of particulate sulfur. We put HMS chemistry into the geoschem model. And so geoschem, we found underestimated HMS at an absolute level during this haze event. And we think this is partly due to the timing of clouds. So the majority of the HMS production is from cloud chemistry. And so minor uncertainties in clouds can lead to large uncertainties in the projection of HMS itself. So importantly, those Shijiazhuang observations were reanalyzed 4 months later after being stored in a freezer, and there was only trace amounts of HMS left. So they weren't really able to detect anything significant anymore. And so this is because, as I mentioned, our hypothesis is that, as I mentioned, HMS can decay over time. There's a reaction where HMS reacts with OH minus to fall back apart. And the lifetime, therefore, of HMS is dependent on the pH. And so at an aerosol pH of 4, there would be a kind of this would have HMS lifetime of around the order of months. And that's where a lot of samples are stored for that kind of duration, and so HMS could fall apart. There's also evidence of HMS falling apart when it's extracted into water and just left out to sit. And so there's been some recent work that's tried adding formaldehyde to the extraction solution to try and stabilize HMS in that way. So we actually were able to find evidence of HMS in the improved network all across the network. So improved samples are stored often for a couple of months between the initial collection analysis. So again, we expect there might be some decomposition, although we don't know the amount of HMS decomposition that occurred. But this here is showing a chromatogram of HMS, chromatogram from the improved network from Birmingham, Alabama. And right here, there's a very clear, clean HMS peak that corresponds to HMS. And the way that we did this is we ran an HMS standard. Importantly, at the general conditions in the improved system, we saw a decent amount of sulfate that appeared from the pure HMS standard, indicating that at least some HMS would decompose to sulfate within the time span of doing the measurement. But then use the relative distances between the HMS peak and the sulfate peak in the standard to identify the presence of HMS going back in different improved samples. And so again, you can see here, there were lots of samples with this HMS peak that corresponded to HMS. And the small peaks here like this are what we saw in the Shijiazhuang samples after 4 months. It was a small peak like this. So what does this look like zooming out? This is the exact spectrum, chromatogram I just showed. And as you can see, the typical system, you look at nitrate, sulfate, and then maybe some other things. But HMS is actually this little teeny peak right here. And so that's typically not looked at. It's this little bit here. So in this spot. So kind of zooming out, we found evidence of HMS in the form of those little peaks like that at 139 of 158 improved sites worldwide. We looked through over 7,000 improved chromatograms, but that unfortunately was not all of the chromatograms for a single year or all of the chromatograms for any single site. So I can't say for sure whether or not sites where we did not find HMS is because it didn't exist or we just didn't look at all of the chromatograms for that particular area. GSChem itself, and this is for wintertime, estimates that up to 35% of the particulate sulfur for some of the United States and over 25% in a lot of regions may be HMS rather than sulfate. And this is not just in the United States, but a major hotspot is, as I said, China, Europe, in the kind of Central Europe, Po Valley area is another hotspot, and we've found HMS detected there. And in general, across all seasons, HMS contributes around, in the model, 7 to 30% of particulate sulfur across the US. Also importantly is wet deposition was the major sink for HMS in the model, and that about 80% of HMS was removed from the atmosphere through wet deposition. So as I mentioned, there's some active chemistry that can happen. HMS can be redissolved in cloud water and fall apart that way. But wet deposition did the lion's share of the of the removal. And so the figure here also is showing the different dots represent the different improved sites where we did detect HMS. The open circles, which are mostly in this area, indicated improved sites where we did not find HMS, but looked at least one chromatogram. And as I said, I don't know if we would have found it if we kept looking. And there have been sporadic measurements of HMS previously, which did find it in other places. And so those are the triangles. So conclusions, wrapping up, are as conclusions. And so new and reanalyzed observations are suggesting that there's a global presence that's pretty ubiquitous of HMS and particulate matter. It remains to be seen how much this is, and again, because there's measurement issues that have still need to be worked out in terms of how much HMS decomposition there is, using the correct or using measurement technologies that can adequately differentiate between HMS and sulfate. GSChem itself suggests that HMS can comprise up to 25% of particulate matter in many polluted regions. And then Lastly, one thing that I think is an important takeaway for people doing measurements is that there are specialized ion chromatography methods that can quantify HMS, but it's important to try and account for this possible decomposition. So doing measurements that are either online or have minimal storage time are important for trying to quantify this. So with that, I will stop and thank everyone and take questions.
Thank you, Jonathan. That was really interesting. You got one question in the chat box from Greg, if you want to go ahead and address that to the whole group.
Sure. So I see Greg's question is, what is the predominant benefit of quantifying HMS in air? Would quantification of HMS primarily benefit air chemistry models? So that's an interesting question. I think that one thing I didn't get into is that the air quality implications of this are that in some areas, we might actually be reducing the wrong compound to try and reduce PM 2.5. So sulfur dioxide, as I mentioned, is the predominant control on sulfate, but HMS can be controlled by sulfur dioxide or formaldehyde. And so in some areas, formaldehyde may be the thing that you want to reduce particulate sulfur. And so there's kind of some big examples of this. One in the Beijing region in China, there's been very significant reductions of sulfur dioxide in the past kind of 15 years, but sulfate reductions has followed much more slowly, especially in wintertime. That might be indicated because HMS in China, especially in wintertime, is formaldehyde controlled. And so that's the red regions here are formaldehyde controlled. And so that means that you'd actually want to go after formaldehyde to reduce this, because 30% or something, or 50, whatever, a large chunk of your particulate sulfur is not responding to your SO2 reductions. A similar thing occurs for East Coast wintertime in the United States. And there's been observations also from the Improved network showing that as SO2 has declined very dramatically since the 2000s, mid-2000s, sulfate in summertime has followed pretty much with SO2 reductions. But in wintertime, it's followed much more slowly. And interestingly, we see that in wintertime in the Eastern United States, HMS would be formaldehyde controlled. And so that's one possible explanation. So the air quality strategies of this are a big importance for why it's important to try to understand what's the fraction of HMS and sulfate. That's a major one. Other questions are kind of remaining uncertainties of HMS. If it is correct that 30% or so in a lot of regions of the sulfur is actually HMS, what does that mean for different deposition pathways or for even climate reflectance, climate implications? For example, HMS hasn't really been studied by itself in terms of what are its optical properties, what are its deposition properties, et cetera. And so if 30% of sulfate forcing is maybe slightly different, that could make a difference as well. And that's an uncertainty that hasn't been looked at. There's a lot of work on HMS in the 1980s, and it mostly dried up after that in the late 1980s.
That's great. Thank you, Jonathan. Thanks so much for that presentation. If anybody has any more questions for Jonathan, please go ahead and put them in the chat. And we'll move on to our second talk for this session. And I'll ask Emma Dambro to go ahead and start sharing her screen. And Emma's going to talk to us about fate and transport of PFAS.
Okay. All right. So I want to start by thanking the organizers for having me. I'm really excited to be here, and thanking my co-authors. It's been a pretty large collaborative work for all the co-authors you see here. So today I'm going to be talking about sumac PFAS development for predicting the fate and transport of per and polyfluoroalkyl substances, or PFAS, in the atmosphere. So just general disclaimer, everything I say is my own opinion, and the work has been internally reviewed and submitted to Environmental Science and Technology. So just start with a little bit of background of PFAS. So PFAS, as I said, are per and polyfluoroalkyl substances. So perfluoro substances are shown on the left, where carbons are completely fluorinated, or polyfluoro on the right, where you still have some hydrogens remaining, but it's largely fluorinated. They're found in a wide variety of industrial and consumer products, and therefore have really been found throughout the environment in different animals, plants, and other ecosystems. So there have been many published studies looking at individual PFAS and demonstrating the impacts on various organs and systems. We're going to be focusing today on the Cape Fear River Basin. So PFAS have been found in the Cape Fear, which is shown on the map on the right in blue, flowing from south of Durham into Wilmington. And of course, this is North Carolina, and it's recently been shown in Wilmington residents' blood as well. We're going to be focusing on a PFAS manufacturer shown on the map, located immediately adjacent to the river. And I should note that not all of the PFAS in the river is from this PFAS manufacturer, but we are going to be focusing on one particular PFAS, which is a proprietary compound of this manufacturer. The reason we're focusing on this location is for a variety of reasons. First, because there's been so many measurements, it's been measured in the water, in the river, as I said, for over a decade. NCDEQ has been started making measurements surrounding this facility, and it's also relatively isolated from other potential PFAS air sources. So the nearest known source is known air source is about 250 plus miles away. So it makes a really good case study for looking at the air transport and fate from a manufacturer. And we know there's strong evidence that there's air transport and deposition contaminating water wells in this area. So shown in the middle in pink is the Chemours facility. And as I said, their property immediately abuts the Cape Fear River, which is flowing from top to bottom, north to south in this view. And NCDEQ has been testing for PFAS in private wells surrounding the facility, and they're color coded by the amount of PFAS. So a green box is a non-detective PFAS, or all PFAS less than 10 ppt. Yellow is intermediate amount on the scale, and then red is if you have Gen X, which I'll get into in a little bit, greater than or equal to 140 ppt. And what I really want to point out in this plot is that it's really complex. So you can have green boxes right next to red boxes, as you see immediately north of Chemours facility. And the other thing I want to point out is that a lot of the high detects in yellow and red fall along the prevailing wind direction, shown with the black arrows here and the windrows on the bottom right. So there's also been high levels of these PFAS shown across the river and upstream from the facility. So areas that are not hydrologically connected to the facility. And North Carolina Department of Environmental Quality has taken measurements at these 5 locations shown in blue here and confirmed deposition of Gen X from air. The influence of air emissions have also been corroborated with qualitative plume dispersion modeling with AirMod by NCDEQ. So we know that there's air emissions from the site, and we already have some measurements indicating that they are depositing at least very near field. And so we have 4 main research objectives with this work. The first is to develop a model of the fate and transport of PFAS air emissions from this facility in North Carolina, and we're going to term that model CMAQ PFAS. Next, we're going to evaluate the predicted deposition of one compound Gen X against measurements taken by NCDEQ. We're going to quantify the AMA air concentration and deposition flux of PFAS with our model, and then we're going to assess the importance of PFAS physical chemical properties in order to determine how complex does our model really need to be. So we're going to start with emissions, because emissions are really of course, the starting point and one of the most difficult parts of this work, because manufacturers do not need to report their emissions because they're not regulated by EPA or states typically. But this facility in North Carolina gave us a detailed air emissions report for 2017 based primarily on mass balances and not on direct measurements of stack emissions. They identified 53 total PFAS, reported a total emission of about 109,000 kilograms in 2017. What this plot is showing is the emission rate in kilograms in 2017 on the y-axis, bin by carbon number on the x-axis. And the reason we did that is because a lot of times PFAS are considered short chain or long chain, with long chain being more, typically more bioaccumulative and more of a concern for toxicology reasons, while shorter chains are maybe less focused on. And we colored them by functionality with the idea that a terminal functional group is going to be more accessible to oxidation and reaction. So kind of giving you a sense of the reactivity of these compounds. And so the plot on the left shows all of the compounds, but 3 compounds make up 84% of the mass. So to give you a better look at the rest of the carbon numbers, we removed those 3 compounds shown on the far right. And you'll notice that they're typically very small. And so the larger compounds with about C3 and higher are typically a little bit more complex with both internal and terminal functional groups. And I'll show that on this slide, where I've picked a handful of compounds and showing some of the physical chemical properties. So there really aren't a lot of measurements. So we used EPA's QSAR OPERA model, which is just the quantitative structure activity relationship. So you tell it the structure and it estimates these various physical chemical properties. And I'm showing CSAR, which is a measure of volatility on the y-axis, and Henry's law on the x-axis. And I just picked a few representative compounds to give you an idea what they look like. You'll notice that most mass, based on the CSAR, should really heavily favor partitioning to the gas phase, although condensation to airborne particles and cloud droplets, as well as deposition of surface waters, is still important to consider based on the Henry's law. The other thing I want to point out with this slide is that a lot of these compounds are relatively large and complex, like this PSE, PVE in the middle. So a lot of different types of functional groups that could be interesting in the atmosphere. So we developed CMAC PFAS. We put 26 explicit compounds with mass greater than 45 kilograms per year into the model and lumped the rest into an other PFAS species with mole-weighted physical chemical properties. And then we implemented reductions based on controls that the facility was implementing throughout 2018. We centered our domain over the facility, shown in pink here, and ran it at relatively high horizontal resolution, 1 kilometer. We ran for the entire year of 2018, again, because that's when measurements were taking place. And I'm going to show 2 different scenarios. The first is a base case scenario, and the second is where we assume all acyl fluorides are emitted as carboxylic acids. And I will explain that in much more depth in a minute. So the very first thing we wanted to do was just evaluate our model based on the existing measurements. And NCDEQ, as I said, was making deposition measurements very near field. So 5 locations within 3 kilometers of the facility, and that's shown the x-axis. The y-axis is showing the daily average deposition in nanograms per meter square per day. The measurements are in pink X's and the model results are in green circles. Note that there are no measurements on the facility in Chemours on the far left or in Fayetteville, the nearest urban area, on the far right. We just put those on there to kind of bound our measurements. And what you see is that the base model is doing a really good job. We're really happy with this result. I should say this is just for 1 compound. It's the only compound that NCDEQ measured that was also emitted by the facility. And again, they are very near field. So the amount of evaluation that we can do on the model could certainly be improved. So now I'm going to show you some of our model results. So this is the ambient modeled air concentration and deposition. So the air concentration is on the left in micrograms per cubic meter, and it's been by the distance from the facility. And what you'll notice is that it's really high. It's really quite high near the facility. So 8.5 micrograms per cubic meter is the maximum annually average air concentration in our domain. But it's the y-axis is on a log scale, so it decreases very rapidly as you move away from the facility. The same is true for the deposition on the right plot. So this is a non-linear color scale. You'll notice the orange color goes from 0 to 60, while the bluest color goes from 600 to 250,000. So really a wide variety, or really a wide kind of range of deposition, concentration, or deposition, again, really high right near the facility and decreases rapidly with distance. And again, these are model results. We do not have any ambient data to evaluate against. And so the biggest implication from this work is that about 5% of the total PFAS is predicted to deposit within our domain. So I'm showing the rate in kilograms per year on the y-axis, and the black bar is emissions and the gray bar is deposition. So you can see that emissions are really high and deposition is relatively very low. And when I say high and low, they're all relative. We don't have any toxicology understanding of these or any measurements to kind of bound them, but so it's all just a relative term. So what this means is that the rest is transported further into the US and beyond. So you're going to be seeing these compounds much farther downwind, and that's already been observed with concentrations in the Arctic and remote oceans. So now I'm going to talk about one specific compound to give you an idea of the deposition and the importance of structure on deposition. So what I'm showing on the right is this HFPO dimer acid anion, and that is what, when people measure GenX, so when the NCDEQ is measuring GenX, this is the compound they're measuring it. When they're measuring it in the deposition samples or in the river, they're measuring this anion, and then the functional group I'm talking about is highlighted in pink. However, when we look at it for it in the air, it's going to be the protonated carboxylic acid form shown in the middle top. And the other compound it could be is this acyl fluoride shown in the middle bottom, which is known to hydrolyze to the carboxylic acid form rapidly in bulk phase water, but we don't know if that's possible in the atmosphere. So the reason we're focusing on this one compound is because we know that there's this kind of, it's more of a family instead of one compound. So we need to understand the play between the two and the different physical chemical properties that are going to affect its behavior. And this is a compound of particular concern in North Carolina, and is something that is being regulated by NCDEQ. And so, as I said, the structure, so if we want to understand what people are measuring in water, and we want to understand where it's coming from, we have to look at both this carboxylic acid and the acyl fluoride. And we have to look at their fate. And so on the left is the carboxylic acid, which is a semi-volatile compound. And the right is the acyl fluoride, which again can hydrolyze rapidly to the compound on the left, potentially. And you'll notice that the volatility is fairly different, and as is the Henry's law. So the carboxylic acid compound is much more soluble. And you can see this effect on the deposition. So the acyl fluoride on the right is only removed by gas phase dry deposition, whereas the carboxylic acid on the left is removed much more readily by gas phase dry deposition. And the numbers on the bars are the fraction of emissions deposited within domain. So it's much more removed by the gas phase dry deposition, and it's also removed by the gas phase wet deposition as well as particle wet and dry. So you can see that effect of the Henry's law and the volatility on its removal processes. So even though these compounds are nearly identical, it's really important to understand which compound you're actually modeling to try to understand the actual fate of this. And so that's why including chemistry and dynamic partitioning in a model like CMAC is really important for capturing the correct atmospheric behavior. And so I wanted to show, so I showed this one compound, the GenX, but there's actually 11 of these acyl fluorides that are emitted from the facility. And so there's 11 compounds that can potentially undergo that, that transformation to the carboxylic acid, which is gonna have a really big effect on the removal. So here I'm showing the percent of emissions deposited within about 150 kilometers. So again, that's our domain size, bin by the carbon number on the x-axis. And you'll notice that when we assume that they're all acyl fluorides in brown, versus when we assume that they're all instantaneously converted to carboxylic acids in the blue, not only are they, again, removed significantly by wet deposition or by dry deposition, you also have a larger component of wet deposition due to that increased solubility. And you'll notice that some of the bars have an increase in deposition and some don't. Where they don't, it's just because there's not an acyl fluoride in that carbon number group. So assuming acyl fluorides convert rapidly to carboxylic acids increases the total deposition of individual compounds. And this deposition shifts from dry deposition to include wet deposition as well. So again, just understanding not only the exact compounds you're trying to measure, but also their reactivity in the atmosphere and what they're going to be transforming into is really important for understanding what you're going to be measuring and finding when you look at deposition. And so we can look at the effect of the total overall deposition. So we increased individual components deposition by going to the carboxylic acid. So we can look at the total suite of PFAS and how that affects the total deposition. And we see that it's not, it doesn't really increase it. So again, the black bar is the rate in kilograms per year of emissions. The gray bar in the middle is the bar I already showed you in deposition. The new bar on the right is when we increase the deposition by assuming carboxylic acids. You see, it doesn't increase deposition that much. And the reason for that is because, as I showed you earlier, 84% of the emissions is from those three relatively small, really volatile, really insoluble compounds. So that's really driving the emissions and the lack of deposition. So the main conclusions I want to go over quick is that we have a new state of the science model, CMAC PFAS, in order to look at the modeled air transport and fate of PFAS. The second is that we found that only about 95% of emissions are transported outside of our domain. And the third is that including chemistry and dynamic partitioning is vital for capturing the correct atmospheric behavior. And I just want to make a quick plug of what else we need to do this work because it's really a budding field. The first, if we want to look, so we looked at one facility, if we want to look at more facilities or across the United States, we really need ambient air and deposition measurements. Those are really lacking, partially because concentrations and deposition are relatively low. So you need pretty low limits of detection. The second is emissions inventory. So as I said, a lot of these places aren't required to report their emissions. Having physical chemical properties is really valuable because as I showed, the difference between an acyl fluoride and carboxylic acid makes a really big difference in your deposition rate. But we kind of have a handle on that with these QSAR models that we are using. And the fourth is toxicity, because we don't know the toxicity of any individual compound. So it's hard to figure out which compound we want to focus on. And the one I really want to emphasize with this group is that ambient air and deposition measurements are really lacking and would be really beneficial for understanding the scale of the PFAS problem much better. And with that, I'm happy to take any questions.
That was great, Emma. Thank you so much. Doug, you'd asked a question in the chat. Did you want to follow up on that since we have a few minutes?
I just wondered if is there any information available about atmospheric lifetimes?
Yeah, so for some of these compounds, yes, for others, no. So for example, if I can go back to this slide, a lot of these compounds are, or I'll go back to this one. A lot of these compounds are actually CFC replacements. So for example, this top compound, this TFE, tetrafluoroethylene, there is quite a lot of information on the atmospheric lifetime. And it's on the order of days. So a lot of these smaller compounds are days, but for something like this PSE, PVE in the middle, this larger complex compound, there's not very much information on what the lifetime is going to be. And that's a lifetime against oxidation.
Okay.
And then in the chat, Chris just asked, how do you specify the phase of the 26 emitted compounds, i.e. gas versus particulate matter, as well as PM size distribution?
Yeah, so we emitted them all in the gas phase, and the reason is coming from this plot where the C star, the vapor pressure, is really very high. So we emitted them all into the gas phase, and that is certainly a source of uncertainty, but I think based on what we know, it's a pretty good assumption based on the vapor pressures.
Okay, great. And Jill just asked, what type of monitors are people using to measure PFAS?
So most people there are some deposition measurements. So NCDEQ is doing those deposition measurements, and they have both bulk, wet and dry, and then they have some wet versus dry, and they're using mass spectrometry techniques to measure the PFAS. But they're mostly targeted. They're not looking for, they're not looking to see what they see. They can look and see what specific compounds they're looking for. On the gas phase side, there are people trying to use mass spectrometry techniques, but I think the main measurement methods have been passive samplers or filter samplers, so offline techniques.
Okay, great, thank you. And I'll just give a quick plug for all the PFAS fans out there. There is a session tomorrow. It's actually in subsession 1 at 11:35. John Offenberg is going to be giving a presentation on the beginnings of a wet deposition monitoring at 4 NADP sites. So if you're interested in PFAS, definitely check that out tomorrow. All right, thank you, Emma. That was pretty amazing, a lot to unpack. We'll go ahead and move on to our 120 talk, which is Amy Sullivan with CSU. And she's going to be talking about a method to measure and speciate amines. Amy, are you there and ready to share your screen?
Yep.
Great.
Okay. Hi, everyone. Today, I'm going to present the details of a method to be able to quantitatively measure and speciate amines in ambient aerosol samples. So just to give you an outline of the talk, first I'll give you a little background on reduced nitrogen and amines. Then I'll give you some details on our measurement method. And then we'll look at some ambient results. In particular, we'll look at measurements of amines from samples collected in Rocky Mountain National Park and from biomass burning source samples. And then I'll finish up with a summary. So ambient reactive nitrogen is considered to be a mix of organic compounds that contain nitrogen as well as inorganic compounds. Now this nitrogen can be in both the aerosol and gas phase, as well as it can be in reduced and oxidized forms. So just talking about the aerosol phase reduced nitrogen, this is predominantly considered to be made up of ammonium and amines. Now amines, of course, is routinely determined in aerosol composition measurements, ammonium is. However, amines really are not. However, there is evidence that amines are in ambient air and that they may be very important. So the most common ambient amines are considered to be low molecular weight aliphatic amines with the carbon numbers of 1 through 6. And so as I'm showing in the red box here, this would include amines such as the series of methylamines and ethylamines. Now, in addition, there's considered to be 4 main sources of ambient amines, and these include industrial combustion, biomass burning, animal husbandry, and from the ocean. And so this means that there are locations where amines could be very important. In addition, if amines really are in the ambient air, this would mean that the traditional cation separation methods that are used to measure ammonium could potentially have overlap with amines that are not being accounted for. And so therefore, the goals of what I'll be presenting today is we wanted to be able to develop a chromatography method to discretely measure amines in ambient aerosol samples. So here, just to show you some evidence for being able to measure amines in the ambient air, here I'm just showing you a comparison of chromatograms taken from an ambient sample collected from a filter sample collected in the Central Valley of California, which is shown on top, versus that of a typical cation standard. And so as you can see, as I've indicated here by the green arrow, is that we are able to see an extra peak clearly between the ammonium and the potassium. So if we go one step further and we show a number of different standards for amines, along with the cation standard shown here at the top, you can see that it's able to confirm that this extra peak belongs to amines. However, you see that with using this traditional cation column, which is a Dionic CS12A column, you can get significant overlap of the ammonium with some of the amines, which includes methyl and ethylamine. In addition, you can see that there are amines that overlap with other amines, and this includes methyl and ethylamine, as well as overlap of propyl, diethyl, and trimethylamine. In addition, depending on what the size of the ammonium peak is, it would be also able to overlap with dimethyl and allylamine, and the same could be true with potassium with propyl, diethyl, and trimethylamine. And so therefore, we tried to extend this method to see if we could get better separation of the amines with the cations. However, even just trying to extend it or changing from isocratic to gradient elution, we're not able to get further resolution. However, it turns out that there are 3 columns available from Dionics that are specific to amines. And so we did evaluate all of these. These include the CS17, 18, and 19 columns. So for the CS17 column, we're never able to fully get separation of the inorganic cations with ammonium. For the CS18 column, we were able to develop a method that could separate them. However, this column only comes in the 2 millimeter diameter format. And so this actually really limits your sensitivity in being able to measure amines, which I'll illustrate a little bit later. However, it turns out that the CS19 column is really a great option, as you can both optimize the separation and it comes in this 4 millimeter size. And so that's the separation I'll be talking about today. So it uses simple eluents of deionized water and 10 millimolar methane sulfonic acid, or MSA. The complete runtime is 2 hours, and that's because it needs to include 3 steps. And that includes an isocratic elution at 0.3 millimolar MSA to get off the +1 charged inorganic cations. Then we have a linear gradient up to 6 millimolar MSA for the amines. And then that's followed by an isocratic elution at 6 millimolar MSA to get off the +2 charged inorganic cations. And it turns out there's really 2 keys to using this method. The first is being able to adjust the suppressor current, since you need to use that really low concentration first step of MSA. And this not only helps to stabilize the baseline, but it also helps in the longevity of the suppressor, especially in the detection of the +2 inorganic cations. In addition, in order to maximize the detection of the amines, you also need to use a large sample loop injection. And this is really where having the format of a 4 millimeter column comes in, so that it can handle this larger volume injection. And we found that using a 1 mil sample loop was optimal. And so overall, by using this method, we get a limit of detection of less than approximately 1 nanogram per meter cubed for the various amines. So here, just showing a calibration chromatogram using this new amine separation. And so what you can see is that we're able to achieve good separation of the inorganic cations and the amines. And overall, we can now quantify 18 different amines. This includes the series of methylamines as well as all the isomers of butylamine. So now if we go back and we rerun that sample that we originally looked at from the Central Valley of California on this new amines column, what you can see is you get a very different chromatogram. And so as I've indicated here now with the green circles, all these extra peaks that you're observing in the ambient sample are coming from amines. In addition, as I've indicated by the green arrow, the largest amine is due to dimethylamine, which comes out just after potassium. So now you'll also see here that the amines are generally much smaller than the inorganic cations. However, all these small peaks really are real, and I can illustrate that here, making a comparison of an ambient sample versus a blank sample. And so here in the top, I'm showing an ambient sample collected from Rocky Mountain National Park here in Colorado. That's, of course, a very clean, pristine area as it's a class 1 area versus a blank sample on the bottom. And so what you see here is that now you're seeing a number of very small amine peaks, but they're only being observed in our ambient sample and not in the blank. And so overall, we can say that we have a very sensitive method that can discretely and quantitatively measure the amines. So now we can take a look at some more ambient data in detail. So looking here further at the Rocky Mountain National Park samples, we collected a daily low flow rate nylon filter from mid-July through mid-October. And so here I'm showing the time series of the different amines, and the different colors just represent the different amines. And so what you see here is that the largest contributor comes from ethanolamine in red, methylamine in blue, diethanolamine in green, and dimethylamine in black. And so what you also notice is that it appears that we have a constant background of the amines of approximately 0.02 micrograms per meter cubed. Now, at the same time, we were making measurements, weekly filter measurements at a site located east of Rocky Mountain National Park in Greeley, Colorado. This site is much closer to agricultural sources, being surrounded by a large number of feedlots. And so here what you see is that we have a noticeably higher contribution from those same 4 dominant amines. In addition, as I've indicated here by the red circle, we see that the highest concentration is observed for the sampling period starting on August 30th. So now if we go back to our data from Rocky Mountain National Park, you'll see that, as I've also again indicated by the red circle, is that here as well, we see the highest amine concentration being observed for that period following August 30th. Now, in addition, for our samples at Rocky Mountain National Park, we were able to analyze them for levoglucosan, whose data I'm now showing at the top of the plot. Now, levoglucosan is, of course, an anhydrous sugar produced from the combustion of cellulose, and so therefore it's considered to be a biomass burning marker. And so what you see here is that the time series of levoglucosan follows along with the amines, and you see that the sum of the amines actually increased about 0.04 micrograms per meter cubed when our site was being impacted by biomass burning, with a noticeable increase in those same 4 dominant amines. Now, in addition to these samples, we were also able to analyze biomass burning source samples. And so here I'm showing the data for these, for the contribution of the different amines, and I have the data applied by, the data segregated by the type of fuel involved in the burn. So we have the data for the burns involving leaves starting on the left, then branches, grasses, and needles. And so first what you notice is that you see a variability among the different amines across the different fuel types. But in addition, as you can see just by the difference now in the color scheme we're seeing here, is that in this case, the dominant amines are actually dimethylamine, which is in black, and or trimethylamine, which is in pink. And so what this suggests is that we see differences in the contributions of the amines from fresh smoke versus aged smoke, as well as likely controlled laboratory burns versus wildfires. And this is interesting and also would require more investigation. However, in addition, because these biomass burning source samples were on quartz filters, we were also able to analyze them for total nitrogen. We did this using a Shimatsu total organic carbon analyzer with total nitrogen module. And so here I'm showing the contribution of the various nitrogen containing species to the total nitrogen for the biomass burning source samples. So we have in red the concentrations for nitrate, in light blue, nitrate, in green, ammonium, and then in dark blue, the sum of the amines, and then in gray would be the other nitrogen species. And so what you see here is that although the contribution for the amines is often higher than ammonium, we still see that altogether, ammonium and amines only contribute about 3 to approximately 10% of the total nitrogen. And so it's really the other nitrogen species that are still dominating, suggesting that although biomass burning is a source of amines, it is likely a minor source. So in summary, we can say that we have developed a robust method using a Dionex CS19 column, which allows for direct quantification of inorganic ions, as well as 18 different amines in ambient aerosol samples. From looking at our analysis of ambient samples, we saw that there were 4 dominant amines, and they appear to be ethanolamine, methylamine, diethanolamine, and dimethylamine. And we saw this to be true for both the Rocky Mountain and Greeley samples. In addition, at both these sites, we saw that we observed about a constant background of amines that increased in the presence of biomass burning. From our biomass burning source samples, it does appear that biomass burning is a source of amines, although it looks like it might be a minor source. And so overall, by using this method and from the analysis of our filter samples, we do have evidence that amines are present in ambient air. In addition, it appears that if you're not accounting for them, there could be the potential to overestimate concentrations of sodium, ammonium, and potassium. And so if we just go back to that original filter sample that we analyzed from the Central Valley of California, if you were just to compare the chromatograms from using a traditional cation method versus this new amine method, you would have overestimated those 3 species by more than 10%. So thank you.
Great job, Amy. So we have about 5 minutes or so for questions before our next talk starts. So does anyone have any questions for Amy? I don't see any in the chat box. You can unmute yourself and just ask if you have anything you want to discuss or have questions for Amy. Okay, Stuart asked, is this the major component of organic nitrogen deposition?
Yeah, so that's a great question. So we haven't run any of precip samples for amines yet. So it's hard for us to say if it looks like that's the case, although we can definitely see that in terms of just looking at how much total nitrogen the amines make up, that it's generally about 3 to 5% from our ambient filter samples.
Okay, Greg Weatherby asked, are you suggesting that the air in Greeley was affected by the biomass burning near the park or vice versa?
Oh, it was actually long-range transported smoke that was coming in that summer. So we were actually getting fires that were coming in from Washington. And so it looks like that although they were both being impacted by the biomass burning, there was probably additional local sources that were also increased on top of that in Greeley as well.
And a thumbs up from Greg. Okay. All right, Vid asked, what's the speculation on the identity of other nitrogen containing species? I guess that was on your previous bar chart.
Oh, sure. So those are, we suspect are more organic nitrogen. Now, in order to be able to really get a more detailed analysis, you'd have to do more mass spec work. But we have seen from some work that we've done looking with HPLC and hooked up to mass spec that a lot of times you can get nitroaromatics are very important, especially in biomass burning impacted samples.
Okay, great. And I think that probably answered Greg's other question too. that he tagged on with it. Greg, did you want to follow up any further on that?
No, that's cool.
Okay, I'll take that as a no. All right. Well, that was great, Amy. Thank you so much. Really appreciate the presentation. And we'll go ahead and move ahead and get a few minutes ahead of schedule. Our next two talks, we have one north of the border and then one south of the border. So that's kind of cool. And up first, we have Michaela Donovan from the University of Calgary, and she's going to talk about atmospheric NO2 and Delta 15N in moss. So Michaela, are you there and ready to share your screen?
Yeah, I'll just get it started here. Okay, so yeah, I'm going to talk about atmospheric NO2 and Del 15N in a wilderness area, and this is in Alberta, Canada. And just before I get started, I'll point out the background here. This is actually where the study took place, so you can keep this landscape in mind. So some background on vehicle emissions in the ecosystem. Vehicle emissions contribute nitrogen compounds to the air, which I'll get into on the next slide. And this can alter ecosystems. So nitrogen pollution can impact the ecosystem through things like changes to the nitrification and denitrification rates. It can cause increased productivity and ultimately decreased biodiversity. And that's because some plants can outcompete others. So this graphic on the bottom just summarizes that. So there's the vehicles and they contribute these nitrogen compounds to the air, and these can deposit onto the vegetation. Okay, so nitrogen in the ecosystem. So the nitrogen gets into the air through nitrogen and oxygen combining in internal combustion engines, and that can produce nitric oxide. Then that nitric oxide will combine with oxygen to produce nitrogen dioxide. So it's really the nitrogen dioxide and the nitric oxide, which is referred to as NOx, that we're really interested in. And that's because the nitrogen in the atmosphere that's just normal that we breathe is not available to the plants, whereas the nitrogen dioxide and nitric oxide are. So essentially with the vehicles, they're increasing the amount of plant available nitrogen. Okay, so the next part is the isotopes, and this is how we look at the source of the nitrogen. So the isotope 14N is what's abundant in the natural environment. And then in the presence of vehicles, the isotope 15N is slightly increased. So when we compare those two, we look at the Del 15N, and Del 15N compares the ratio of the rare isotope, which is this 15N, to the abundant isotope, and it compares it to a standard. So essentially, if the Del 15N is higher, that suggests that there's a bit more of this 15N, which is commonly contributed by combustion. And if the Del 15N is lower, there's less of that rare isotope. So we can presume that there's less vehicles that are contributing that rare isotope. Okay, so this study took place in the Kananaskis Valley, so I'll show you where that is. Here's a map of North America with the United States and Canada here. So these two Western provinces, I'm going to zoom into those so you can kind of see where we are. So here's the US-Canada border. This province on the west is BC, and then Alberta is here. And here's the Rocky Mountains. So the study took place on the eastern slopes of the Rocky Mountains, where this little square is, and now I'll zoom into that. So this white rectangle here shows the study area. And then you can kind of see this highway here. So this is Highway 1, which is the Trans-Canada Highway. It runs from the west of the country to the east, and it's a major, major highway. And this red line here is where the study took place. It's called Highway 40, and it's smaller, just one lane each way. So this is the north part of the valley, and this is the south part of the valley. And the white arrow represents the flow of traffic. So because that Trans-Canada Highway is in the north, the vehicles enter the north, and then so the gradient is expected to be from the north to the south. Further to the west along the Trans-Canada is some of these inner mountain towns, and then the west part of the country. And to the east is this city here, Calgary. It's a major city. So that's why we expect most of the people to be entering from the north. Although there is an entrance at the very bottom of this valley, it's not expected to be used as much as this northern one. So Kananaskis Valley is a protected area. You can kind of see here there's a couple names of parks. So there's provincial parks, wildland parks, wilderness areas, recreation areas. These all have different goals and different objectives in protecting the area, but nonetheless, they're all protected. So that's really the interest of the study, is this is a protected area and we're trying to preserve the pristine nature here. So the big question for the study was, is nitrogen pollution from vehicle emissions observable in the ecosystem in the Kananaskis Valley? So we wanted to see if we could detect any signs that there was nitrogen getting into the ecosystem in this protected area. So we were expecting two different things. We looked first within the valley. So this was just a spatial scale. So we expected traffic to increase from south to north. So north is where that Trans-Canada Highway was. So we expect most of the vehicles to be in the north. Then because of that gradient in traffic, we expected nitrogen dioxide to increase moving south to north. So as you move towards that major highway at the top, there should be more nitrogen dioxide in the air because there's more traffic. And then the Del 15N values in Moss should increase in the same way. So in the south, that ratio should be lower because there's less of that rare isotope. Whereas you move towards the north where there's more NO2 expected and more traffic, there's more of that rare isotope contributed to Moss from vehicles. So that ratio should be increasing. And then we looked at among years. So this was the temporal scale. So we first expected for traffic to vary over the years. So the study took place from 2003 to 2019, and we expected some increases, some years with more traffic, some years with less. And I'll point out specifically 2013, there was a flood in the Kananaskis Valley. It was pretty major. It impacted Calgary, the city downstream, and the golf course was closed and the road was closed. So we expect a lot lower traffic this year. And then the Del 15N values in Moss were expected to vary with traffic among the years. So in any given year that the traffic was high, the Del 15N values should be high as well because there's more of that rare isotope. And then same with 2013, because we're expecting less traffic in 2013, we should have less of that rare isotope that is contributed to by combustion in that year. So the study took place from 2003 till 2019, and it was the data was collected for the first 2 weeks of September. This was done by a field course. So every year the students went out in the summer or in the September to collect the data. And so we used the data from that study. So the first step was to look at the traffic. So we used these automatic TrafX counters, placed them at the sites of interest, and counted the amount of vehicles that went by. The purpose of this step was just to see if there truly were more vehicles in the north than in the south. Then for nitrogen dioxide, we use these passive samplers. So it allows the nitrogen dioxide to diffuse into the tube, and there's some treated filters in here, collects the nitrogen dioxide, and we can analyze that. So these passive samplers were placed at the sites of interest, just 5 meters from the roadside along that highway that I showed, the one in red. And then for Moss, we collected the Moss samples from the locations that these passive samplers were out. They get dried in an oven, ground to a powder, and then they're analyzed for that isotope ratio. Okay, so then the first result was that there was more traffic in the north. So this plot shows the amount of vehicles per day plotted against northing. So northing measures the meters north of the equator. So the smaller number here would be the furthest south, and as the number gets larger, you're moving further away from the equator. So this graph shows that in the south part of the valley, there's less traffic, and then moving north towards where that major highway was and the common entrance to the valley, there's more vehicles. So that confirmed our first prediction and allowed us to continue on with the other analyses. Then this shows the nitrogen dioxide. These are the passive samplers that we use, just as a reminder. So this is how we got this data. And this shows the NO2 residuals, which I'll go back to, plotted against northing. So again, this is the furthest south, so the more remote part of the valley. And this is the furthest north. Now, I have residuals here because this data includes many years because the data was collected from 2003 to 2019. So what we did is we plotted the NO2 with the year, and then we took the residuals to remove that variability that's expected for years. And then the remaining variance is plotted against northing. So this plot shows that in the south of the valley, where there were fewer vehicles, there's less nitrogen dioxide. And then as you're moving north towards where that entrance was, there's more nitrogen dioxide in the air. So that was consistent with what we saw with the traffic. Okay, and then for the Del 15N in Moss, we saw it was higher in the north as well. So this is the same thing. This is the residuals. So we plotted it with year first took the residuals so we could remove the variability in year, and then plotted it against northing. Again, this is the south part of the valley and the north near the entrance. And again, as you can see, there's a less of that del 15N ratio in the south than in the north. So as you're moving north, the ratio is increasing, which means that that rare isotope is getting more and more, which suggests that vehicles are contributing more of that rare isotope because of the fractionation. Okay, so what did we see in the valley? Well, we thought that traffic would increase from south to north, and we saw that there was more traffic near the north where the entrance was. And because of that, we thought that nitrogen dioxide would increase from south to north, and we saw that as well. So that followed the same pattern as traffic. And then finally, we thought that the del 15N values in moss would increase in the same way, south to north, and we saw that as well. So it suggested that because there's more traffic in the north, there's more nitrogen dioxide in the air, and there's also more of that rare combustion isotope. Okay, so this is moving on to the temporal scale. So this is the amount of vehicles per day plotted against a year. And so as you can see, from 2003 to 2012, it's pretty consistent. There's not really any changes between like the amount of vehicles in the valley. 2013 then drops significantly, and then it starts to increase after that. And it's interesting because I mentioned the flood at the beginning. So this is kind of what we're seeing here. So there were a lot less vehicles. And then it starts to increase after that. So then because we saw that pattern, we could then plot the MOS, the del 15N against the years. So this is the annual del 15N ratio plotted against a year. And it's a little bit variable. There's some up and down here. 2003, I should point out, there was, when the Clean Air Act was, they were putting the different phase changes in 2003, I think 2004 had the new standard. So we presume that that's what we're seeing here, is that's why the del 15N ratio drops so suddenly. But aside from that, it's kind of hard to see a pattern, although there is variability between the years. It's not overwhelmingly increasing or decreasing. But if I go back to the plot with traffic over the years, it looks kind of similar. So it's consistent up until 2012 and then increasing. And aside from 2003 here, it's relatively consistent and then increasing. So then because those two were so similar, we thought, let's plot the del 15N ratio against the actual traffic numbers. So then that's what I'm showing here. So this is the annual del 15N ratios plotted against the amount of vehicles in each given year. And then 2003, sorry, that's when the policy change happened. So this was a significant outlier. So with 2003 removed, we can see that there's less, when there's fewer vehicles, the ratio of the rare isotope to the abundant isotope is lower. So there's less of that rare isotope. And then as you're increasing the amount of vehicles, there's more of that rare isotope. So this suggests that the vehicles were in fact contributing that rare isotope to MOS. So what did we see among the years? Well, we thought the traffic would vary from 2003 to 2019, and we saw that. So it was kind of consistent up to 2012, and then it dropped at 2013 and starts to increase. And then we thought whatever we see in traffic, we should see in MOS. And we saw that as well. So it was a slightly different, but there was the drop in 2013 and then the increase. And furthermore, when we plotted the del 15N values against the actual vehicle numbers, we saw that it increased. So that rare isotope from combustion increased with the amount of vehicles. So in conclusion, we're saying that the nitrogen deposition from vehicles is impacting the ecosystem. So just to reiterate, this is a wilderness area. So you can see there's just one lane each way, and it's remote and it's pretty untouched. But we are seeing that there's an impact of these vehicles in the ecosystem. This has been seen before. So increased NOx in the del 15N and MOS has been observed in the Grand Canyon National Park. But Kananaskis in the beginning in 2003, I think they saw like something like 500,000 vehicles a year. Now it's over 1 million. But the Grand Canyon National Park sees about 4 million, I think. It's the second most visited national park in the States. So it's kind of surprising to say we can see this even at really, really small, relatively small traffic volumes. And so just to conclude, why do we care about this? Well, increased nitrogen can alter plant quality and quantity, and this might attract species. So if species are being attracted to roadside verges, this could potentially increase or decrease their success. And these impacts could be kind of brought throughout the landscape as these species move around. So for an example, Mitchell et al., one paper, they looked at something that they called the ecological trap, because vehicles were putting these attractant pollutants in, like nitrogen. More species, specifically the monarch caterpillar, was being attracted to these areas, but it's an ecological trap in that not only the nitrogen is getting into these plants, but other toxins from emission. So they're coming because it's going to be a more biomass and high quality food, but there's also some impacts to this. Okay, that's everything. Thank you very much.
Great job, Michaela. So we have a couple questions in the chat box. First, Stuart asks, did you measure the Delta 15N and NOx itself? And what about ammonia from catalytic converters?
Yeah, that's a good point about the ammonia. So there's a couple studies that, and I think in the literature it's a little bit inconsistent too. So we decided that because there is so much of the ammonia that comes from almost the same amount of nitrogen deposition of ammonia comes from nitrogen dioxide. So we just had to kind of have that as an assumption, because we aren't familiar with the complex relationship between the isotopes and the Delta 15N. So yeah, we only measured the nitrogen dioxide and just had to assume that that's what we were seeing.
Okay. Eric's asked, is there nitrogen fixation in these mosses, and will that affect the Delta 15N?
So I'm not even a biologist, so some of these things are like a little bit hard for me to grasp. So I think there might be some, and so there are a lot of assumptions with the study. And one thing we wanted to do was actually use moss bags this summer, and so we're not grabbing. We have really controlled variables with the moss, because these are assumptions of what's going on with the moss, even when it grew and how long it's been there. But we couldn't do this study with the pandemic. So yeah, that could be a potential as well.
Greg asked, is the moss perennial or annual? And is the Delta 15N cumulative in the moss over time, or are these truly annual measurements from the moss?
So they were perennial, so they're all year round, but we also have a lot of snow in this area. So that's something we were considering as well. And if we could expand the study, we would start looking at how the snow might affect these things. And if we could kind of control, that goes back to the moss bags. Are we measuring just the 2 weeks that the passive samplers are out, or when we're collecting the moss, are we looking at the whole year? And how does the snow actually impact if the deposition is on top of the snow? What's really happening here? So I guess we don't really know if these are truly annual or if they're just from the 2 weeks. So hopefully we can expand on that. And there's a lot more research to be done in the area.
Great. So Mike asked, did you perform any transects perpendicular to the road to get a sense of the distance impact from the road, or the distance of the impact of the road?
So for the field study where the students collect the data, they do a control that's like 50 to 100, but we didn't do any transects at all. There's plenty of studies that look at how far into the forest, but we thought the gradient down the highway was kind of a strength of the study, because there's still vehicles on the areas that we're seeing less nitrogen deposition. So it's, instead of vehicles and no vehicles, we're seeing a lot of vehicles and some vehicles. So yeah, we didn't do any transects. We just kept it with the gradient down the highway.
Vid asked, are there plans to do plant species studies to determine ecosystem impacts?
Probably not by us. I think we could, so we used 2 species of moss, and there was no difference between the way that they absorbed the nitrogen from our study. So that would be kind of interesting to look at the different plant species. So we don't really have anything, but there's tons of literature, and maybe not a consensus even, because some of them are more negative effects. Some say mortality, and if there's like, for example, coniferous forests, then deciduous take over after that mortality from nitrogen pollution. But there's a lot of positive effects too, like nitrogen is a limiting nutrient, so it increases the biomass. So I think it would be a super long-term study, but we don't have anything in plan for that.
Okay, and one final question. Does the traffic counter differentiate between small cars and large trucks?
No, it just counts exactly what's there. And that's an interesting question, because for the field school, the students actually sit and count for like 1 hour. So we didn't use this data, but they count if it's a recreational vehicle, because there's a lot of camping down here. There's no real like semi trucks or commercial vehicles because it's a pretty remote area. But that's a consideration as well. The traffic counters just count every single vehicle that's going by, no matter what it is. So we just use that raw number.
Excellent. Well, thank you so much. Really appreciate you doing the presentation with us today.
Thank you.
Any further questions for Michaela, please put them in the chat or email with her. And we'll go ahead and move up to our 2 o'clock talk. We welcome in a very familiar face to many of us, Rodolfo Sosa. He's going to talk to us about advances in the evaluation of reactive nitrogen in Mexico City. Hello, Rodolfo, welcome. And you can go ahead and share your screen.
I am going to present an example of collaboration among the academical and governmental institutions in Mexico City, and the collaboration with institutions at the United States, NADP, USGS, and USEPA. I want to take advantage of the first slide in order to congratulate and recognize the effort of the NADP staff in order that the fuel meeting for this year will be successful. In this case, the collaboration with the Mexican government started in 2003. Mexico City, as you know, is located in the center of Mexico, and the Mexico City is surrounded by some municipalities of the Mexico state. All both areas form part of the Mexico City metropolitan area. The most important industries are located in the north, and the dominant winds come from the north to the south. And regarding the precipitation in the area, this precipitation presents different values depending on the altitude and depending on the land use. The higher levels in precipitation are located in the mountains region in this area, and the lower precipitation in the plain area. In the world, you know that Mexico City represents an excellent laboratory for studies about atmospheric pollution. There are a lot of factors that made the designation. In this area, exists more than 20 million inhabitants, more than 5 million vehicles, and is located at an altitude more than 2,200 meters above sea level, and is surrounded by mountains because the city is located in a basin. What about the air quality evolution of the Mexico City metropolitan area? The first studies start during the 80s, and the principal pollutants were sulfur dioxide, total suspended particles, and lead. Which was the reason, because most of the 30% of the industry of the country was located in Mexico City. And during this year, the principal fuel that were used for energy generation was fuel oil. At the end of the 80s, the fuel oil was switching by natural gas, And this was a successful in the reduction of sulfur dioxide total suspended particles. Another pollutant that was present during the 80s and the beginning of the 90s were lead that was used lead tetraethyl like anti-knocking in the gasoline. The reduction of lead started at the end of the 80s, and finally, in Mexico, at the beginning of the 90s, we used unleaded gasoline. But the atmospheric chemistry changed, and now we have the problems of ozone and particles, PM10 and PM2.5. Another important strategy that has been developed in order to reduce the atmospheric pollution in Mexico was to cancel the operation of the refinery that was located inside Mexico, and several industries were located outside. And at this moment, the increase in the traffic, in the number of vehicles is one of the problems, and the population are increasing at this moment. Regarding the SU2 air quality, at this moment we are in a attainment area. We don't have problems with our Mexican air quality standard that is so similar to the United States standard. But the interesting problem that we have at this moment is the acid rain. Acid rain was worse during the 80s and is decreasing, but the problem continues. And of the ions contained in the precipitation, the most important that is originated by the precursor is the sulfate ion. Sulfate is the most important ion comparing with nitrate. And why is the reason? Because we have emission sources of sulfur outside Mexico City. Here, I want to present the network. The network is a very nice effort for part of the Mexico City government. The first network that exists in Mexico was since 1985 for the measure of criteria pollutants in an automatic way. The network is the RAMA by the Spanish name, Automatic Atmospheric Monitoring Network, and regards the nitrogen compounds. At this moment, they are measuring NO, NO2, and nitrogen oxides. And the other network that is very important is operating in working together with the University of Mexico since 2003. Mexico City government is responsible for the sample, and the University of Mexico laboratory is responsible for the analysis. The nitrogen compounds at this moment we are measuring are nitrates and ammonia. Here are some pictures of different locations at the above network are operating in Mexico. Around 30% of the sampling sites are located in semi-rural areas, and the other are located in urban areas. This is the map. This is the map of the air quality monitoring network. More than 40 air quality monitoring stations operates this network and are located at Mexico City and in the surrounding area. The most industrial industries are located in the north. The landfill that now is closed, but both the landfill was operated by several decades, is located at the northeast, and the dominant winds come from the north to the south. This is represent an important results when we are going to show the air quality data and the atmospheric deposition results in the different stations. This is the REDA, the atmospheric deposition network. They collect the samples weekly. The protocols that we use are similar to the national trend networks. We are in touch with NADP, USGS, WMO, and EPA. And these are the sampling sites remember the landfill is in this area. The dominant winds come from the north to the shore. And pay attention in this sampling site that this nest is the station that presents the higher levels of ammonium. The analysis of the data that we are presenting in this meeting is from 2003 to 2019. And we divide in two. One is the part of the ambient air and the other is the weather atmospheric deposition. The ambient air has been measured because it is necessary to evaluate criteria pollutants in order to comply with the air quality standards. And regarding nitrogen compounds, NO2 is the criteria pollutants, but the methodology by chemiluminescence permit the evaluation of NO and nitrogen oxides. Furthermore, nitrogen oxides has been very important for several decades to be evaluated due to role as a precursor of ozone. And Mexico City is an area that presents a big problem of photochemical pollution. Some of the stations, how can you see, this is an example of the rural sampling site. The other is located in the middle of the urban activity. Some results that I am present here are about nitrogen compounds in ambient air, only nitrogen oxides. We can observe that there is a reduction in all the pollutants, NO, NO2, and NOxes, and the behavior is different depending on the region of the city. The north station present the higher values, downtown station the middle values, and the south side the lower. This is affected, this very influenced due to the activity is here, the high industrial activity. The mobile sources are distributed in all the city, and there are a lot of industries located at the north of Mexico City and at the north outside Mexico City. At this moment, this is a very interesting experiment that was observed due to the pandemic COVID-19 period. This is the information about the year 2020 from January to September. This is an average of all air quality monitoring stations, only to represent the behavior of these pollutants. The nitrogen oxides is reduced. At this moment, the last information that we have is until September. Start increasing slowly. And if we compare with the last year, 2019, the behavior of the nitrogen oxides is similar. This is a cycle. During the rainy season, the levels of the atmospheric pollutant are reduced. However, during the pandemic, the reduction in the concentration of primary pollutants were increased due to the reduction of the activity and, of course, in the reduction of the emissions of different industries, but the more strict reduction were applied for the mobile sources. Around of the 50% of the traffic in Mexico City were reduced, and the emissions were reduced in the same ways. Another topic that is very important to talk is photochemical pollution, because in ozone the result was not the same. We continue studying these kind of pollutants during this experiment that is called the pandemic COVID-19 period. Okay, the weather atmospheric deposition is analyzed at the laboratory, the University of Mexico. The red of the government take the sampling at the 16 sites. The UNAM chemical analysis use the protocols for sampling and analysis for NADP and UCPA and participate in the laboratory in the comparison programs with the World Meteorological Organization and the United States Geological Survey. This is an example, this is an average of all the results. Here are presented the principal ion is the ammonium, then the sulfate, both the anion related with the acid rain precursor, sulfate and nitrate. Sulfate is bigger compared to nitrate. This is the reason why we need to continue thinking to solve the problem with sulfur, to solve the problem of sulfur in the soil outside Mexico City. This is the results about hydrogen ion. Consider the 60 sampling sites for wet atmospheric deposition evaluation. The sampling sites are ordered according with the altitude. The higher altitude located in the area of the mountains and in the south present the more acidic values, influenced by the dominant winds that come from the north. And we are going to see that some species that can neutralize the acidity are lower in this area. So this is a multifactorial and complex result. And in the case of nitrate, here appears the lower values regarding the mean, the median, the equal values according to the mean, and the peaks are located at the north. What ammonium? Ammonium is a case that is very necessary to study because most of the study are regarding the nitrogen in the oxidized form, but we need to increase the studies of the nitrogen in its reduced form. Please check the values of the median are located at the source, and the bigger or the higher values are located here at the north areas. Remember that at the northeast is located the landfill, and the dominant winds come from the north to the south. This is the reason why these areas, this station, present the peaks in ammonia. Can you see here? Here. This is the bigger median. And the radio, ammonium nitrate, present the same behavior. The lower values at the south, and these stations located in the northeast, in the downtown at southeast, checking the dominant winds, present the higher values. This is the higher value in these stations and represent a value of 2.4. The value or 2.4 among ammonia and nitrate is similar to other urban areas in North America. We have been in touch with Greg, with Terby in Denver, Boulder, and the results look so similar. Another way to present these results, the ammonium represents the 71% of the nitrogen ions. The nitrogen ions in wet atmospheric deposition at this moment, that's only ammonium and nitrates. It is very important to evaluate the reduced nitrogen in the ambient air. And ammonium represents of the total chemical composition of the rain, the 30%. And the highest values are in the region of high urban and industrial activities, mobile source, the landfill. We need to include the meteorology. As a proposal, in order to evaluate the reduced nitrogen in ambient air, we are proposing that it's necessary to establish a network for ammonia. It's very important the collaboration with people from other countries. I appreciate all the support and help from NADP, UCPA, and USGAs. And this is a proposal using the same sampling size of the network, the automatic network, the atmospheric deposition network, and establish 6 sampling sites for the measurement of ammonia. At the beginning, I planning to prepare some proposal to establish the same kind of equipment that are used in airmon, radielo sampling, and then analysis at the lab, and maybe 1 or 2 automatic equipments. I appreciate the help of John Walker, that I hope that the next year will be working with us at Mexico City University campus in order to install automatic monitoring equipment for ammonia. And the next steps is to collaborate with other urban areas in North America. The example of atmospheric nitrogen chemistry in Denver is very interesting. I remember the last meeting in Boulder, we had the opportunity to visit the site and to reinforce the reactive nitrogen studies in urban environments in North America. 2 days ago, we talked about the CTDEF program of NADP. This is an opportunity to join efforts, not only binational or trinational. We need to join efforts as a global way in order that all the countries take advantage of this very important opportunity that is to be here in NADP. Thank you very much. Do you have any question? I appreciate your time.
Thank you, Rodolfo. That was great to see what you all are doing. Does anybody have any questions? We have a couple of minutes. So for your ammonia monitoring, Rodolfo, what are you thinking that you're going to start doing?
The last meeting of the NADP, I had the opportunity to talk with different people of NADP and was planning to start this year. I visit EPA, Research Angle Park with John Walker. He's very happy. I suppose let's continue being happy to start the monitoring of automatic ammonia. But the pandemic period was a problem in this case. I hope for the next year, when the safety condition of the pandemic will be solved, will be possible to start with the automatic equipment. But we need to continue working with Greg, with David, and with John, and with all people, all my NADP friends, in order to start with the passive samplers, radielo, and analysis at the laboratory, and applying some technologies like the loaders in order to use active sampler, not only passive. But I have a lot of ideas, but the pandemic is a problem at this moment. I hope that the next year we can continue to work. Maybe for the next fall meeting of NADP, we can present some results. And I hope that the next fall meeting will be face-to-face.
I think we all hope that, Rodolfo. I totally agree. So thank you. That sounds great. All right, and we will move on to our final presentation in this session, and that is monitoring spring water nitrate as an indicator of atmospheric deposition by Stuart Weiss. Stuart's with Creekside Science. So Stuart, are you there and ready to share?
Just a brief introduction. I want to thank the organizers, and the pandemic has transmogrified me into Jerry Garcia, for those of you who I know personally. So I'm going to talk about biodiversity conservation in the Bay Area through the lens of the Bay checkerspot butterfly. So the Bay checkerspot butterfly is listed as threatened under the Endangered Species Act, so it has a high degree of legal protection. It lives in this really remarkable habitat, which is a native California grassland, very rare ecosystem, growing on very nutrient-poor soils derived from serpentinite rock. The ecosystem has been, and the butterfly have been studied for decades. The fantastic place to work, And I extend an invitation to anybody who's in the Bay Area in the springtime to get in touch with me, and I'll get you out into the flowers. We noticed back in the 1980s that in the South Bay, which is south of San Jose, when cattle were removed from the grassland, we got this very intensive growth of non-native annual grasses on these very nutrient-poor soils. That led to the loss of butterfly populations. So, empirically, we knew that the cows were really important, and the cows eat grass and leave room for the beautiful wildflowers and for the butterfly habitat. The butterfly relies on a couple species in the grassland that get crowded out by the grasses. And the culprit behind all of this is the atmospheric nitrogen deposition in the Bay Area. We're looking north towards Silicon Valley and it's disappearing into that smog cloud. And back in 1999, I had pulled together all the various threads of evidence and published a paper in Conservation Biology. That paper led to the first mitigation for increased nitrogen emissions from this 600 megawatt gas-fired power plant. And that set a precedent for mitigating for nitrogen deposition impacts because of the Butterfly and the Endangered Species Act. The need for this kept coming up. So this is the north side of one of the butterfly habitats in 2002. And because of a Pacific Gas and Electric cut off the cattle grazing on this area just about this time, this is what the habitat looked like in 2007. So quite obviously a major loss of biodiversity. You don't need to be a botanist to tell this one. So what do we know about the deposition and the composition of our smog cloud, that's almost a daily occurrence? I got together with Andrzej Witnierowicz, who almost everybody here must know. We set up a network of passive samplers, the Ogawa passive samplers across the San Francisco Bay Area. Down here are the South Bay sites. The wind basically blows generally northwest and accumulates all the air pollution down here. And we got results like this. This was published in a paper in 2010 at these sites in the South Bay. Tulare Hill is the dirtiest. Back then it was getting between 15 and 20 kilograms per hectare per year. A little further south at low elevation, about 15. And at the top of the ridge farther south, about 12. A lot of ammonia in the deposition, and then a lot of nitric acid vapor as well. The mitigation for the power plant and for a highway project led to the instigation of developing a regional habitat conservation plan. It gave birth to the Santa Clara Valley Habitat Agency. So it's called the Valley Habitat Plan for short. It's a habitat conservation plan and a natural communities conservation plan. The NCCP part is a California state process. The HCP is a federal process under the Endangered Species Act. The plan was adopted in 2013, about 12 years after the original biological opinion said that they had to do this, which is actually pretty fast for a plan like this. The ultimate goal is to acquire and manage about 46,000 acres for 19 covered species, and 10 of the 9 are reliant on this serpentine grassland. So the nitrogen deposition is a major nexus for this plan. The cost is estimated in 2013 as $665 million over 50 years. I know that sounds like a lot. It's $13 million a year on average, but this is Silicon Valley, where Literally, this is rounding error in the Silicon Valley economy. And one of the policy innovations was a nitrogen deposition fee, a small fee per new daily vehicle trip. It comes out to about $45 per new single family residence. It's a one-time fee. And I don't think this is why housing costs are so expensive in San Jose, but it's going to be generating about $10 million over the 50-year plan. A small part, but a significant part of the funding of this plan. And a big part of it is to maintain the ranchers on the land so we can have the cows and the associated mammalian species taking care of the impacts of the nitrogen deposition. So I want to look a little bit more in depth here. This is the TDEP map for 2012-2014. I really want to thank everybody who worked and developed TDEP. It's an amazing product for the kind of work that I do. So we can see, here's the sites in the South Bay, and you can see that there's quite a gradient from urban San Jose in red down into more rural areas. And when we pull out particular grid cells, we can lay them out in a gradient going from about 13 kilograms per hectare per year in the middle of San Jose, down to about 5-6 kilograms per hectare per year farther south. And see the breakdown of it, that ammonia is really a large proportion of this in the South Bay. Nitric acid vapor is relatively constant across the gradient, and then NOM, or primarily nitrogen dioxide, drops off as we get a little further south. Particulates are a minor component. So the idea is, how do we monitor this over the long run? So we want to look at monitoring spring water nitrate as an indicator of deposition trends. So we know nitrate leaching is a strong indicator of nitrogen saturation. We want to know how much is in the spring and stream waters and how does it vary in space and time. Is there a correlation with deposition gradients? What's the future trajectory of nitrogen deposition? And basically, we want to know, can spring water nitrate be used for long-term monitoring, because it's a relatively simple measurement to do. So serpentine grasslands are riddled with springs. We did some recon surveys across the range. And here, this is just a subset of the springs that we looked at. An example is there's a spring in that grove of trees that's draining the serpentine grassland up above it. A very interesting place where they dug out a cave many, many, many years ago as a water supply. And unfortunately, this cave collapsed just this last year, so this beautiful pool is no longer. This is more typical. Here's the headwaters of a little canyon. There's a seep in a spring right here, and water's coming out. So we ended up picking about 20 sites. Cattle troughs are really good because there's a spring box that's sampling shallow groundwater before there's a chance for any surface interactions. So it's really simple. We go out at the end of the dry season, like right now, grab a water sample, send it off to an EPA certified lab, come back with a nitrate value. So here's kind of the whole data set through 2019. This first site is a domestic well at one of my staff guys' house. But our dirtiest sites have about 5 parts per million nitrate as nitrogen. And our cleanest sites are down about 2. All of these are well above the 1 part per million threshold for nitrogen saturation. And these are as high as I've ever seen in a non-agricultural circumstance. So we can start decomposing this. We find that spatial patterns are positive relationship with northing, i.e. the closer you are to San Jose, the higher the spring water nitrate is, and then lower elevations have higher nitrate and higher elevations have less. So basically the nitrate is higher closer to San Jose and at lower elevations, which is essentially the deposition gradient we measured with our passive samplers. The trend through time's a little murky right now, but I have three sites where we have 5 years of data. And there's definitely big differences between the site and a upward trend, but unfortunately the p-value is 0.08. We're not seeing large changes from year to year, which is good, but there does appear to be an overall upward trend. If we look at what TDEP is telling us in terms of total deposition at the various sites, it's telling us that there has been a slight increase in total deposition. And that's primarily, I'm not showing it here, it's increases in ammonia. So if I keep harping on ammonia in vehicles, there's a reason for that. So let's look at the leaching process and the ecosystem effect. So this is a typical soil profile, if you call that soil, at during the dry season. Measurements of soil mineral nitrogen seasonality. So here in September, at the end of the dry season, we actually have accumulations of ammonium and nitrate. In November, there's a big burst of biological activity and the nitrate really bumps up. But then by February, the plants are much more active and there's a big sink for nitrate and ammonium especially. And then we start seeing the accumulations start again in May. And from this particular study, it was 2 parts per million nitrate nitrogen in the leachate that was below this experimental plot. So the leaching occurs at the beginning of the rainy season when we get enough water. There's a lot of nitrate in the soil and it flushes through. The total annual plant uptake is estimated to be about 20 kilograms per hectare. And when we're getting 10 kilograms per hectare as deposition, it's a substantial fraction of the annual plant uptake. If we look at the soils here, it's a Montera series soil, very shallow, 33 centimeters to bedrock, pretty high saturated hydraulic conductivity. And then you get to the bedrock, we're not sure from the soil exactly what's happening here. But we have these hydrogeologic studies at this large landfill, the Kirby Canyon landfill, that investigated a lot of this because of the potential impacts of the landfill on groundwater. So they measured groundwater velocities. There's 3 different layers that are of interest. There's the alluvium colluvium, where they estimate about 50 to 200 meters per year, and about 92% of the water is flowing through this layer that's about 1 to 6 meters thick. Below that is weathered serpentinite, order of magnitude lower velocities, about 8% of the groundwater is flowing through this 6 to 9 meters thick. And then the unweathered serpentinite is really tight, and the groundwater velocities are less than 1 meter per year, and less than 1% is flowing through these areas here. So we're interested in, when we're measuring the nitrate, how many years of nitrate are we looking at? So I delineated some spring catchments. The flow does follow the topography. So when I did that and applied some Darcy's law, I became a hydrogeologist for a second. We can see that the residence time in years on a lot of these springs is just a few years. So we're looking at sort of an average over, 1 to 3 or 4 year period down here. So we should see responses as deposition changes through time. There's also fracture flow, a lot harder to get a handle on, but that's definitely faster than through the rocks. So in the long run, there's been quite a big reduction in total nitrogen deposition. Here's TDEP from 2000 to 2002. Notice how red it is. Here it is in 2012 to 2014. In large reductions. And if we look at these sample sites, we see the largest reductions are in the other oxidized category, which is primarily nitrogen dioxide. We also see commensurate decreases in nitric acid vapor in blue, but then we see all of the increases in ammonia. This is the story across the United States with big decreases in oxidized nitrogen. This is downtown San Jose nitrogen dioxide. Thank you, Air Resources Board and Environmental Protection Agency for this. But the real culprit in the ammonia is the catalytic converters, where these cars are all emitting ammonia along with the NOx. And co-author on this paper by Mark Fenn and company about it. And we really do feel like this is an underappreciated source of atmospheric nitrogen deposition. The long-term trends are downward for NOx. We don't really have a good, and ammonia is just this big question. It's supposed to stay steady, but it's not really well accounted for in emissions inventories. But we're looking at like another 30% reduction in NOx, but then it eventually levels out. So in summary, serpentine grassland downwind of Silicon Valley is nitrogen saturated from atmospheric deposition. The nitrate levels in the summer base flow are higher than in any other non-ag system I've been able to find. If somebody knows of one, let me know. It tracks the spatial nitrogen deposition gradient. The leaching potential is really high in the early season, and essentially the annual deposition is flushing right through the system because there's no more ability to take it up. The mean groundwater residence times is a few years at most in a lot of these springs. So I think we can track multi-year average deposition very inexpensively by taking water samples. My crew is going out tomorrow to do the 2020 samples, so I'll keep you posted next year. And generally, we're having declining NOx and increasing ammonia, which is the story across a lot of the US. So this ecosystem is just absolutely gorgeous, a major conservation target. And this Valley Habitat Plan and other mitigation projects since 2001 have permanently conserved more than 1,500 hectares of this ecosystem and butterfly habitat. And the major nexus was the atmospheric nitrogen deposition. And the Bay checker spot butterfly thanks us all for finding all of this out. So a lot of agencies and funding support. I really want to thank the scientific community here, kind of untangling that fiendish complexity of the nitrogen cycle in air, soil, and water and ecosystems. And then, of course, I need to thank the Bay checker spot butterfly for leading me down this long and winding road. Thank you.
Thank you, Stu. That was great. Got a couple questions that have come in.
Sure.
And from Doug, asked, are you fairly certain that there is not any significant contribution of nitrogen from mineral weathering?
Yes. Yeah, the serpentinite is a metamorphic rock derived from the mantle, and there's absolutely no mineral nitrogen or organic nitrogen in the rock.
Okay.
Also, when we measure in clean air areas, the nitrate is like below detection limits in truly clean air areas, which are hard to find these days.
Well, Greg just had a nice comment that this is a great case study, and I agree completely. It's really neat to see how it all kind of connects together. And then he makes a pitch for an Airmont site. So there you go, Greg.
Yeah, the more we can understand about deposition in this environment, we actually have a really fantastic, let me just go back. Well, we have a great split plot experiment up on the San Francisco Peninsula. That photo I showed you of the freeway right through a serpentine area. That would be great to be able to isolate the effect of a single road with about 100,000 people. We have passive samplers there, but it'd be great to kind of bring on the really heavy duty equipment and understand a lot of the details.
Thanks, Stu. And Greg said he meant AMON, so that makes more sense since that's an active network. But thanks. I just want to say thank you to all 6 of the presenters during the session. I just think all 6 of these talks were outstanding. And I wish we could all applaud really loudly for all 6 of you. We'll do a virtual applause. And thanks again for all of you tuning in. We averaged somewhere around 100 participants throughout the entire 6 talks, so that was great.